Let's invite up Matt Marshall from the Department of Biology. The topic of Matt's research is the genetics of thermoplasticity in Plantago lanceolata.
Good afternoon. Today we're facing a major problem. The Earth's climate is quickly changing, yet we don't know how organisms will respond to these changes. It's imperative that we have at the very least, plants survive into the future, because all land animals rely on plants for food and oxygen, including you and I. And it's for this reason that I've chosen to study the genetics of thermoplasticity in Plantago lanceolata for my dissertation research. Now, thermoplasticity is simply the way an individual responds to changes in the temperature in their environment. And Plantago lanceolata is this perennial herb that's common around the world. And I've chosen to use Plantago lanceolata for my research because an individual can easily be split down the middle by cutting it, and multiple pieces can be grown. I grow some in a warm environment, others in a cool environment, and measure the difference as the response to temperature. In nature, individuals differ in their response to temperature. And so, for example, here in the red box, I'm showing a low plasticity individual. This individual shows a very small response. When grown under warm or cool environments, it produces lightly colored flowers and has tall, skinny leaves. High plasticity individuals, on the other hand, shown here in blue, respond strongly to temperature. When grown in a warm environment, they also produce lightly colored flowers and tall, skinny leaves. However, in a cool environment, they produce much darker flowers with shorter, thicker leaves. These darker flowers allow that individual to absorb more sunlight and warm up the developing seeds and offspring inside those flowers, and the shorter, thicker leaves help reduce heat loss to the environment. As a result, we often find high plasticity individuals away from the equator, where a greater part of the growing season occurs at cool temperatures, and low plasticity individuals closer to the equator, where warming up isn't as important. To get at the genetics of thermoplasticity, I've crossed low and high plasticity individuals to produce hybrids that have 50% of each parent's DNA. Then I crossed both hybrids to produce 500 grandchildren shown here. Of these grandchildren, some have very low plasticity, some have very high plasticity, but most are somewhere in the middle. Next, I read the DNA of every individual in this family and ask which pieces of DNA do those low plasticity grandchildren share with their low plasticity grandparents. And likewise for the high plasticity grandchildren, which pieces of DNA do they share with their high plasticity grandparents? These are the pieces of DNA that respond to temperature change. The main goal of this research is to identify how many pieces of DNA respond to temperature change and on which chromosomes they lie. Identifying the genetic basis of thermoplasticity will help for other researchers forecast and hopefully mitigate the effects of climate change as we try to feed a growing population in a changing climate. Thank you.